Hi guys, so in this video we are going to be addressing the common misconceptions about chapter 5, which is all about macromolecules. So one common misconception is that our organic molecules are linear and two-dimensional instead of being three-dimensional. So when we see our molecules, we usually see them like this, represented in a linear fashion, just like we see this glucose. And the reason that we represent our molecules linearly is because it's going to simplify complex structures. So a linear representation strips down the complexities of molecules and is only going to show the essential components, such as its bonds and atoms. So a linear representation simplifies our molecules. So, for example, glucose, like we see here, it has a formula of C6H12O6, but like we see here, it's represented as a straight chain, even though it's normally found as a six-membered ring in solution, in an aqueous solution. But this simpler linear version, not only is it easier to draw, but it's also easier to help identify the functional groups that are found in it. But in reality, our molecules, they're normally 3D structures, and these 3D structures, they're super dynamic. And they're dynamic because they are not rigid. Right? So our molecules, they are not rigid. They can change their shape due to the rotations around their bonds, movements of atoms, and interactions with the environment. And this flexibility that molecules have, it allows them to adopt different conformations, which is necessary for their function in biological systems. And an important note that I want to make is that bonds in organic molecules, they allow rotations unless they are stabilized. So they do not rotate. So there is no rotation when there are double or triple bonds. Bonds, when they are double or triple, they don't allow rotation because they have something called pi electrons. And these pi electrons, it's going to create a planar structure and it's going to lock those atoms in place. But single bonds are also known as sigma bonds. And they are able to freely rotate because the electrons that are bonding each other, they are symmetrically distributed. And a symmetrical distribution of electrons is going to allow for rotation. But when we have double or triple bonds, that structure is more rigid and locked in place and stops rotation. And another important point to make is that even though we normally see molecules in a 3D ring form, they can be seen in a linear form, even though it is rare. In the linear form of glucose, we have single bonds between the carbon atoms that allow free rotation, which make this molecule flexible. But when we come up here to our functional group, this one, our carbonyl group, we see that we have a carbon that is double bonded to an oxygen. And specifically, this is an aldehyde. And with this double bond, we have no rotation allowed because of the double bond that is found between that carbon skeleton and this oxygen atom. But in all of our other bonds, they are all single bonds, and these bonds, they are allowed to rotate freely. When we look at glucose in its ring form, which is the most common configuration, we see that rotation is restricted because the oxygen atom inside of the ring and the carbon atoms together, they form a stable cyclic structure. And cyclic structures, they're going to stabilize glucose and limit bond rotations within the ring. This is not a hard and fast rule. We can still exhibit rotation in cyclic structures, but it just has other criteria that has to be met, but that is completely out of the scope of this class. So for this class, we can kind of associate cyclic rings with no rotation, but that is not completely true. But overall, this linear form is going to allow free rotation in 5 of the 6 carbon skeletons. And there's going to be no free rotation allowed at the carbon to oxygen double bond at our aldehyde group. But in the ring form, there is normally no rotation due to the rigidity of the cyclic structure that it has. Another common misconception is that proteins only have one level of structure, or that they are simple linear chains of amino acids. So normally, we're going to see our proteins represented as straight chains like we see here. And that's because it's a lot easier to show and understand their basic sequence when they're represented that way. That linear representation of proteins, it's used because it highlights that primary structure of our proteins. And remember that the primary structure of proteins is just the order of amino acids in a chain. Of amino acids in a chain. But in reality, our proteins, they're going to fold into specific 3-dimensional shapes that are essential for their functions. And a 3-dimensional shape is started to be seen at the tertiary structure of proteins. So the different shapes that are formed by the 4 different structural levels of proteins are our primary structure, which is the linear sequence of amino acids. And remember that these amino acids, they are connected by peptide bonds. And these peptide bonds, they were formed through dehydration reaction. But after we have our primary structure, we move on to our secondary structure. And the secondary structure, it occurs when that primary structure starts to fold into very simple shapes like this alpha helix shape. And it's going to start folding into this shape because of hydrogen bonds between that backbone of atoms of the amino acids. But at the secondary structure, it is still 2-dimensional. And hydrogen bonding begins the folding. So when we get to the tertiary structure, we have the overall 3-dimensional shape of our protein. And this is formed by the interactions between the R groups of amino acids. So the shape of our tertiary structure, it is formed due to the interactions between the R groups of our amino acids. And the interactions, it can include our hydrogen bonds, ionic bonds, disulfide bonds, which are very strong covalent bonds, and it can even include hydrophobic interactions, just to name a few. But when we get to our quaternary structure, which is our 4th and final structure, that's going to occur when 2 or more proteins, they come together to form a functional protein. So a quaternary structure is a functional protein with multiple subunits, which is what we see here in hemoglobin. It has multiple different subunits, which form its quaternary structure. And a very important note that I want to make is that not all proteins require the quaternary structure to be active. Some proteins are actually functional once they achieve their tertiary structure. So function begins at the tertiary structure for select proteins. So all proteins, they must reach at least the tertiary structure to have the potential to be active. And in the tertiary structure, the protein's final shape, it's fully formed. And that shape of the protein is what's going to determine if it can be functional or not. But the quaternary structure, on the other hand, the quaternary structure is critical for multiple subunit proteins, where the interactions between our subunits, that is necessary for them to be able to function fully. And if we don't have that correct assembly of our subunits, then the protein cannot function properly. But the main point of this is that a protein can be active at the tertiary state when and only when it is a single subunit, but multiple subunit proteins must be in that quaternary structure to be active. So the last misconception that we're going to talk about is the idea that nucleic acids are only limited to DNA and RNA. So to start, I'm going to define a nucleic acid, and a nucleic acid, it is just defined as a large biological molecule that are essential for storing and transmitting genetic information. So a nucleic acid is a large biological molecule that is essential for storing and transmitting genetic information. And they also participate in various cellular processes. And these nucleic acids, they are composed of nucleic acids, nucleotides. So our nucleic acids are composed of nucleotides, and all nucleotides have three key parts. They have a sugar, they have a phosphate, and they have nitrogenous bases. So if anything has these three things, it is considered a nucleotide, which is what makes up our nucleic acids. So when we're looking at our DNA and RNA molecules, what we see is that the sugar in our DNA molecule is called deoxyribose, and the sugar in our RNA is called ribose. And our phosphate group, it's here. It's what's forming this helical structure, double-stranded on the DNA and single-stranded on our RNA. And then we have our nitrogenous bases, which are these. And our nitrogenous bases, we can call them nucleobases. And the nucleobases that we have, they're going to depend on whether we're looking at RNA or DNA. But there's 5 total. We have cysteine, guanine, adenine, uracil, and thymine. So normally, when we see nucleic acids, we see them represented as double bonded stranded helices for DNA, which is what we're seeing here, double bonded strands in DNA. But when we're looking at RNA, we see a single strand. And that representation of our nucleic acids, it emphasizes the role that they have in storing our genetic information, since our DNA and RNA are our stores of information. And using simplified representations like this, it allows the focus to be on the essential components like that sugar phosphate backbone and the nitrogenous bases. It's going to help to visualize base pairing in DNA and the single-stranded nature of RNA. But in reality, our nucleic acids, they are about more than just our DNA and RNA. We have a lot of very important nucleotides, which include ATP, which is adenosine triphosphate, and it includes many others, such as cyclic adenosine monophosphate or CAMP. And there's so many important nucleotides. And these nucleotides, specifically ATP and CAMP, they play very important roles in cellular function. And when we're looking at the structure of ATP, what do we see? We see sugar. And the sugar that we find in ATP is actually ribose. And ribose, it is a 5-carbon sugar. So we would have a carbon here. 1, 2, 3, 4, and then here's our 5th carbon. But we also see that ATP contains this phosphate group. And not only that, but it has a nitrogenous base over here. And in the case of ATP, it has adenine as its nitrogenous base. So overall, as long as a molecule has a sugar, a phosphate, and a nitrogenous base or basis, it is going to be considered a nucleotide. And by extension, it is a nucleic acid derivative. And these are just the 3 key points of this misconception for chapter 5. We need to remember that our organic molecules, they are dynamic. They are not super rigid. And normally they are 3-dimensional. We have to remember that our proteins, they have multiple structural levels, and each of their levels is essential to determining their function. And we also need to remember that our nucleotides, they are not limited to DNA and RNA. They do so much more than that.